Hello, professors and classmates. My name is Zhang Wan'an, and I'm going to present you my term project, Myocarditis Preventing mRNA Vaccine from Site-Selective Nanoparticles. We will start from the introduction. Myocarditis describes the condition when the heart muscle becomes inflamed. The inflamed heart muscle or myocardium is not capable to function normally, so it may not pump blood as effectively as the original myocardium. Shortness of breath, chest pain, and arrhythmias are common symptoms for myocarditis. According to clinical statistics, there are 0.3 to 5 cases of post-mRNA vaccine myocarditis per 100,000 vaccinated people, and higher incidence is observed when the second dose of vaccine is administered. Although most patients spontaneously recover after chest pain syndrome, hospitalization or even death are still possible consequences of myocarditis. The potential mechanism of post-mRNA vaccine myocarditis include innate immunogenicity and cross-reaction of antibodies against COVID-19. Single-stranded RNA is the substrate of endosomal receptor toll-like receptor 7 or TLR7 and TLR8, which can trigger the production of pro-inflammatory cytokines. Other possibility is related to the antibodies against the virus. The antibodies that can neutralize the virus can cross-react with the myocardial alpha-myosin heavy chain, which is structurally similar to the spike protein of the virus. The immune system may thus elicit inflammatory response. In order to solve the problem, site-selective lipid nanoparticles, or LNPs, are used to precisely deliver mRNA molecules to a specific part of the body. By targeting vaccine lipid nanoparticles to CD205 on the migratory antigen-presenting cells, or APCs, and subsequent transport to lymph nodes, antigen-specific immunity can be enhanced with effective antigen presentation. Not only can anti-CD205 surface-modified lipid nanoparticles enhance the immune responses, but also prevent the transfection of mRNA molecules at heart, which may lead to potential inflammation. The mRNA of anti-inflammatory IL-10 is delivered to myocardium to prevent tissue inflammation by inhibiting the activities of cell signaling proteins. Local rather than systemic anti-inflammatory effects of IL-10 on myocardium can be achieved. So here is the experimental design. 2 lipid nanoparticles are injected into mice intramuscularly. First, anti-CD205 lipid nanoparticles encapsulating the mRNA of spike protein can interact with dendritic cells and be transported to lymph nodes for antigen presentation. Second, myocardium targeting lipid nanoparticles encapsulating the mRNA of IL-10 can deliver anti-inflammatory IL-10 to myocardium to elicit local anti-inflammatory effects to prevent myocarditis. The cooperation of dendritic cells targeting lipid nanoparticles containing mRNA X vaccine and myocardium targeting lipid nanoparticles containing mRNA encoding IL-10 makes it able to reduce the risk of inflammation locally at myocardium, while maintaining the immune response required for antigen-specific immunity. Now let's talk about experimental suggestions. The lipid nanoparticles are prepared using nanoprecipitation by microfluidic mixing. This is the microfluidic device. And this is the mRNA molecules. The ionizable lipids can be protonated at acidic condition, which can interact with negative charged mRNA with electrostatic force. And as the polarity of solvent increases, the pegylated lipid come together and form spherical lipid nanoparticles. The pegylated lipid at the shell of lipid nanoparticles can prevent proteins from attaching on lipid nanoparticles and thus reduce the possibility of clearance by the immune system after opsonization of lipid nanoparticles. For anti-CD205 lipid nanoparticles, anti-CD205 antibodies are linked to the surface of lipid nanoparticles by the esterification reaction of carboxylic groups on the antibodies and hydroxyl groups on pegylated lipid with carbodiamide. For myocardium-targeting lipid nanoparticles, cardiomyocyte-specific binding peptides are ligated to LAMP2B proteins. The amine groups of the LAMP2B proteins, which enhance cardiomyocyte uptake, are linked to the hydroxyl groups on pegylated lipids on the surface of lipid nanoparticles by the succinic acid base NHS. For in vitro transfection efficiency, cardiac muscle cell and dendritic cells are seated in transparent 12-well plates. Then 24 hours later, lipid nanoparticles are prepared, and the amount of lipid nanoparticles that contain 1 microgram of mRNA are used to co-culture with the cells. After another 24 hours of incubation, the fluorescence signal from EGFP expression is evaluated with flow cytometry. For in vivo transfection efficiency, lipid nanoparticles encapsulating the mRNA of firefly luciferase are injected intramuscularly into the mice. After 24 hours of inoculation, luciferin is injected intraperitoneally into the mice. The organs are collected for ex vivo bioluminescence imaging using IVIS. For in vivo humoral immune response, anti-CD205 lipid nanoparticles encapsulating the mRNA of the spike protein and myocardium-targeting lipid nanoparticles encapsulating the mRNA of IL-10 are injected intramuscularly into the mice. Serum samples are collected and examined the amount of spike protein-specific antibody with ELISA kit. For in vitro prevention of myocarditis, serum samples are collected and examine the amount of cardiac troponin T, a specific indicator for myocardial cell damage, with ELISA kit. Heart tissues are collected, then stained with H&E. The potential toxicity of lipid nanoparticles are measured using blood chemistry and H&E stain of the main organs. And here is the expected results and discussion. For in vitro transfection efficiency, the fluorescence signal of EGFP can be detected by flow cytometry when the mRNA is successfully translated into the protein. Anti-CD205 lipid nanoparticles and myocardium-targeting lipid nanoparticles lead to selective transfection efficiency. The EGFP expression is higher in dendritic cells when the anti-CD205 lipid nanoparticles are used, because CD205 enhances endocytosis of lipid nanoparticles. In contrast, the EGFP expression is higher in cardiac muscle cells when the myocardium-targeting lipid nanoparticles are used, because the proteins on the myocardium-targeting lipid nanoparticles can boost the targeting effect. The results of transfection efficiencies towards two different cell lines demonstrate the potential feasibility of in vivo site-selective mRNA delivery. For in vivo transfection efficiency, the firefly luciferase mRNA will express a luciferase protein which emits bioluminescence in the presence of the substrate, luciferin. The highest radiance of luciferin happens at the spleen when anti-CD205 lipid nanoparticles are used, because the spleen contains dendritic cells carrying lipid nanoparticles after CD205-mediated endocytosis. Also, the highest radiance of luciferin happens at the heart when myocardium-targeting lipid nanoparticles are injected. These findings confirm that two-site selective lipid nanoparticles are able to accumulate at the desired sites. For in vivo humoral immune response, the production of spike protein-specific antibodies determines the effectiveness of the vaccine. The groups with the injection of anti-CD205 lipid nanoparticles encapsulating the mRNA of the spike protein show the highest IgG concentration in the serum samples. This is because CD205 expressed on the surface of dendritic cells is responsible for endocytosis and antigen presentation. The humoral immune response is therefore stronger. For in vivo prevention of myocarditis, cardiac troponin T or cTNT is a specific indicator for myocardial cell damage. The groups with the mRNA of spike protein show the highest cTNT levels because high level of spike protein specific antibody is a potential reason for myocarditis. Myocardium targeting lipid nanoparticles encapsulating the mRNA of IL-10 can induce anti-inflammatory effects at cardiac muscle cells, which leads to a cTNT level as low as the control group. In addition, microscopic inspections of tissue sections of myocardium stained by H&E indicate significant inflammatory infiltration in the groups with mRNA spike protein. On the other hand, the injection of both anti-CD205 lipid nanoparticles and myocardium targeting lipid nanoparticles shows no obvious inflammatory infiltration. These results suggest that the anti-inflammatory effects can locally triggered by IL-10 at myocardium without reducing the production of spike protein-specific immunoglobulin triggered by anti-CD205 lipid nanoparticles. For biosafety, no dysfunction of organ is observed, showing that the lipid nanoparticles formulations do not exhibit significant systemic toxicity. In conclusion, anti-CD205 lipid nanoparticles encapsulating the mRNA of the spike protein interact with CD205 on dendritic cell. Humoral immunity is therefore enhanced. Myocardium targeting lipid nanoparticles can deliver the mRNA of IL-10 to cardiac muscle cells and elicit anti-inflammatory effects. The proposed therapy features local anti-inflammatory effects at myocardium while maintaining the immune response required for effective vaccines. So this is my term project. Thank you for listening.